Welcome back, dear students. Today we are going to be talking about cell transport mechanisms. Cells, as we have seen in the introduction to physiology, are made from organelles that are bound and contained within a plasma membrane. The plasma membrane, PM, is the structure that determines the boundary that differentiates the inside of the cell from the outside. Although it's imperative there be adequate exchange and communication between the inside and outside of cells. The fluid on the outside is called the extracellular fluids, ECF, and is very different from the composition of the internal fluid of a cell, which is called the intracellular fluid, ICF. Therefore, the plasma membrane allows for transport and exchange, but also maintains the differences between the two sides. We are going to concentrate mostly on the role of plasma membrane proteins as transporter in the body and the various mechanisms by which they move molecules from one side of the plasma membrane to the other. There are two categories of protein transporters. First is protein channels, and second, protein carriers. Protein channels are well named. They are much like little water-filled channels, forming a passageway that directly links the intracellular fluid to extracellular fluid. The narrow diameter of protein channels restricts the passage through them to small-sized molecules, mostly water and ions, potassium, sodium, chlorine, and calcium. Electrical charges lining the either channel may restrict the movement of some molecules. Therefore, they can be very specific as to what they allow to travel through them. This mode of transport is very fast, much faster than protein carriers, because there is no need for the binding of any substrate as in protein carriers, so the substrate can flow right through. Which of the two molecules in the drawing to the left will get through this protein channel and why? Gated protein channels. Some protein channels are gated and need to be opened before allowing ions to travel through them. The protein channels that have gates that can open or close are called gated ion channels. First type, chemically gated channels. There are three types. First type, chemically gated channels triggered by specific ligands, chemicals, that can open or close the channel. Second type is voltage gated channels triggered by electrical changes across the cell membrane to open or close the channel. And the third type is mechanically gated channels. They triggered by distension or physical force applied to the membrane to open or close the channel. The second type of protein transporters are called protein carriers. These never form a direct or continuous passage between the ECF and the ICF. They have a binding site like enzymes and will only transport specific molecules that match this site. Once the molecule binds to the site, the protein carrier undergoes a conformation shape change. It can rotate or close at one end while it opens at the opposite end, thus carrying the molecule across membrane. This mode of transportation is slower than protein channels as they need to bind the substrate and change shape while moving substrates. A perfect example is glucose movement across membranes. You may have heard plasma membranes described as selectively or semi-permeable membranes. This means that some molecules can get across and some molecules cannot. The membrane composition determines which molecules move across. Permeable molecules can cross membrane by any method. Impermeable molecules cannot cross cell membrane. General factors influencing molecule permeability. Although the components of a plasma membrane can vary, the properties of a given molecule will have a large effect on whether it passes through the plasma membrane easily or if it needs assistance or if it cannot pass it at all. Below are the three main factors that determine whether a molecule can pass easily directly through the plasma membrane or not. First is size of molecule. Smaller molecules can more easily pass through than larger molecules. Second is polarity or lipid solubility. Lipid soluble molecules pass through more easily than polar molecules. And third is charge of molecule. Uncharged molecules pass through more easily than charged molecules. The permeability of a molecule can be influenced by all three of these factors, not just one. There are two ways a molecule can be transported across a cell membrane: passive and active transport. Passive transport. Passive transport doesn't require energy or ATP. It just involves movement down a concentration gradient. There are three basic categories of passive transport in the body. First is diffusion, second is facilitated diffusion, and the third is filtration. Diffusion is the net movement of molecules from an area of higher concentration of that molecule to an area of lower concentration of that same molecule. In other words, it's the movement of the molecule down its concentration gradient. There are some factors that affect the rate of diffusion. Size of the molecule. The smaller, the faster. Distance. The shorter, the faster. Temperature. Warmer, faster. Surface area of membrane. Greater, faster transport. Thickness of barrier. Thinner, faster. And steepness of concentration gradient. Greater, the faster. As we continue in physiology, all of these factors will be revisited in the various organ systems. A second type of transport is facilitated diffusion. Some molecules that are polar or too big to use simple diffusion to get across a membrane can use protein carriers to move down the concentration gradient. This requires no energy. This is different from simple diffusion because the molecules must bind to the membrane carrier, as discussed above. So in this way, it needs the help of a protein carrier to move down its gradient across the membrane. And the third is filtration. Filtration is the net movement of water and solids across a membrane due to the force of hydrostatic pressure HP. Hydrostatic pressure can be defined as the force of a fluid on the walls of its container. The water in a cup has hydrostatic pressure, as the water exerts a force on the cup containing it. The next, active transport. Active transport always requires energy inputs, always requires ATP. There are three types: primary, direct active; secondary, indirect active; and vesicular transport. Active transport requires the input of energy from ATP. This ATP is required because molecules are being moved up or against their concentration gradient. It's proteins that move these molecules against their concentration gradients. It's important to realize that creating and maintaining this equilibrium across the membrane in the body is very important and useful. Again, this requires the input of energy from ATP. The movement can be of one or more substances across the membrane. Uniport. When a protein moves only one kind of molecule, it's termed uniport. Co-transport. When a protein transporter moves more than one molecule at a time. There are two types. First, symport moves the molecules in the same direction, and antiport moves the molecules in the opposite direction. Okay, primary active transport, direct. In primary active transport, energy from ATP is directly used to transport molecules against their concentration gradient. The best example of this is the sodium-potassium pump, or sodium-potassium ATPase. This is a membrane-spanning protein carrier. And the next type is vesicular transport. Vesicular transport is used to move large macromolecules or large quantities of a molecule across the plasma membrane. There are two main forms of vesicular transport. First is endocytosis. Endocytosis is branching material into the cell. Inward vesicular transport. There are three general kinds of endocytosis. The first type is pinocytosis, cell drinking, relatively unselective, whereby intracellular fluid is transported into the cell. Second, phagocytosis, cell eating, which is a process whereby cells engulf a particle of or another cell into a much larger vesicle. Third type is receptor-mediated endocytosis. Next type of vesicular transport, exocytosis, releasing material from the cell. This is the outward transport of contents via vesicles. This type of transport is used by many cells to secrete or release large molecules or a large amount of a molecule. Intracellular vesicles fuse with the plasma membrane, then release its contents into extracellular fluid, ECF. This process requires energy, ATP, and calcium, and also involves other proteins.